In 1996 was chlorea, an acute diarrheal infection caused by ingestion of food or water contaminated with bacterium called Vibrio chlorea. This disease kills the
Tetanus is a
individual
disease
within
caused by
hours.
bacterium Clostridium tetani, which is found on rusted nails and surfaces. It is characterized by painful muscle spasms and can be prevented by regular vaccinations.
Polio is a highly infectious viral disease that mainly affects young children. It causes difficulty breathing and sometimes leads to death. The virus is transmitted person to person through fecal-oral infection or sometimes through contamination of food and water. The last time this disease occurred naturally was in 1979 in the US and has been controlled through the poliovirus vaccines.
These diseases have caused many to suffer and some to eventually die. Today, however, there is hope. Vaccines. Vaccines have eliminated the suffering of many and prevented many to do so in the future.
After the creations of vaccines, there came an even better creation: recombinant vaccines.
1965, Dr. Baruch Blumberg and a group of scientists went around collecting blood samples in Indonesia's people. He studied the samples, and when he did, he made a remarkable discovery. Together with his team, Blumberg discovered an unusual antigen from this blood sample of an Australian Aborigine which had turned out to be the cause of hepatitis B. Using the sample, his colleague, Dr. Irving Millman, invented the hepatitis B vaccine. It was named the very first anti-cancer vaccine because it prevented chronic hepatitis infections, resulting in the prevention of primary liver cancer due to HBV. A couple of years later, a similar vaccine was invented, but with a little difference. It was a recombinant vaccine.
But what is a recombinant vaccine? A recombinant vaccine, also called recombinant vector vaccines, are vaccines that use viruses or a bacterium to introduce microbial DNA of cells to the human body. In other simpler words, these types of vaccines consist of a portion of pathogenic virus fused with a harmless bacterium. This is then injected into the human body.
Recombinant DNA technology was first invented in 1979 by two biochemists named Herbert Boyer and Stanley N. Cohen. Their achievements represented an advancement upon the primary technique invented by Paul Bury in 1972 for inserting viral DNA into bacterial DNA. Present today, we have a few genetically engineered vaccines. Using the bacteria cell E. coli, they successfully showed that genetically engineered DNA molecules may be cloned in foreign cells.
There are different kinds of vaccines, such as recombinant protein vaccines, live recombinant vaccines, and lastly, recombinant DNA vaccines.
The recombinant proteins in the vaccines allow the immune responses focused against protective antigens and are used to fight against hepatitis B. Live recombinant bacterial vaccines are able to stimulate the immune system with natural infections and have intrinsic adjuvant properties effectively. DNA vaccines, which consist of non-replicating plasmids, can induce strong long-term cellular immune responses. Generally, the purpose of vaccines is to train the immune system to recognize and combat pathogens that could harm, most usually being a virus or bacteria.
How is it made? In nature, viruses latch onto cells and inject their DNA into them. Scientists have researched and studied many ways on how to manipulate this process and use it towards the human race. The creation of recombinant vaccine starts off by taking a non-pathogenic sample of a virus/bacteria, then fusing it with another bacterium cell. First, there is an isolation of a bacterial plasmid found in the bacterium. The newly formed plasmid is then inserted into the new bacterium. A small dosage, now considered a vaccine, is then injected to the human body. The cell then replicates until it's detected by the body's immune system, which then responds with antibodies to fight against it, also becoming familiar with this type of virus/disease for the future. So what are the benefits of this vaccines? Similarly, scientists continue to research this technology. They include providing a much more efficient and strong immunity towards the bacteria/virus compared to regular vaccines. Advantages also include being represented as an ideal and safe, heat stable, and easy to administer. Preferably single dose and capable of inducing broad immune system with lifelong memory both in adults and infants. Are there any disadvantages? Sadly, there are. Genetically engineered subunit vaccines are most costly to manufacture compared to conventional vaccines. This is because the antigen must be first purified to higher standards. Furthermore, each vaccine must be subjected to being FDA approved, thus leading to extensive testing, resulting in more time and more money required.
All in all, recombinant vaccines have done a lot for us. They've created a new passageway for scientists to help humanity and will continue to do so in the future.